Hello and welcome to the Australian Biocommons webinar series, where we aim to share useful information about the latest digital techniques, data, tools for the life science community. Each month we hear from our local and international peers who present a bioinformatics topic that we hope will support Australians to deliver their best environmental, agricultural and medical research. My name is Melissa Burke and I'm the Australian Biocommons Training and Communications Officer, and I will be your host for today. We particularly appreciate those of you joining us live today. You will have the opportunity to ask questions via the Q&A function on your dashboard, and these will be addressed at the end of the presentation. The session will also be recorded and you'll soon find it on our YouTube channel, along with recordings of previous webinars and workshops. We also hope that you'll keep in touch to hear about future webinars and other events via the channels listed on the screen. Before we start today, we would like to take a moment to acknowledge the traditional owners and the custodianship of the lands on which we meet today. In my case, this is the Gittabul people of the Bundjalung Nation. We pay our respects to their ancestors and their descendants who continue cultural and spiritual connections to country. We recognise their valuable contributions to Australian and global society. Today, we are thrilled to welcome Justin Bedo to speak to us about unifying package managers, workflow engines, and containers, computational reproducibility with Bionics. Justin is the Stafford Fox Centenary Fellow in Bioinformatics and Computational Biology for Rare Cancers at WeHi. He was awarded a PhD from the ANU in 2009 in machine learning. His PhD investigated novel machine learning algorithms to solve bioinformatics problems arising in plant breeding and cancer genomics. After his PhD, he obtained postdoctoral experience at NICTA and IBISC Grenoble, Paris, as well as industrial research experience at IBM Research Australia. His interests span machine learning, diagnostic genomics, and bioinformatics. Welcome, Justin. I'll now hand over to you to lead the presentation.
Thank you very much for the kind introduction. It's my pleasure to talk to you today about Bionics, which is my tool for managing bioinformatics workflows. So in this context, a workflow is some bit of analysis that may be comprised of several steps, and you perform this on some inputs producing some final analysis. And the goal is to make this as reproducible as possible. While there are many tools that do this currently available, Bionics is quite unique in its approach to this problem, and as a result can give very strong reproducibility guarantees. And furthermore, it takes a very functional approach to this problem, which allows the specification of workflows to simplify down to function compositions, which, as we'll see later, makes it very easy to specify and manage pipelines and workflow definitions. So to begin with, to motivate why workflow management is needed in the first place, we observed that bioinformatics workflows tend to be quite long-running and error-prone. And as a result of this, it's very important to be able to both handle errors properly and also resume a partially complete workflow after resolving any errors that were encountered. Because they're long-running, we also need to be able to parallelize the steps involved in executing a workflow and distribute them potentially across multiple machines. And many analysis is comprised of more than just a simple sequence of steps, which means that a simple bash script does not scale to them well, and that tooling is needed to increase the reproducibility of our workflows and analyses. In my mind, there are three core problems that need to be resolved to produce a reproducible workflow and analysis. The first of these is to manage software versions and their various dependencies. If you think of typical solutions that are currently available, one might think of package management systems like Condo and say Debian's DBKG and apt-get system. These bits of software allow you to manage software, install them into an environment, and update them as well. Some of these package managers also have a specific repository for bioinformatics software, such as Condo, which provides many common tools that are used in bioinformatics, which is convenient. The second problem is to manage computational environments. So being able to install software into an environment is one thing, but you may need to have multiple environments within which to execute your analysis. And this is especially important in the case of requiring different versions of certain bits of software, as you may not want them all exposed in the same computational environment. In many cases, people resort to containerization these days, using tools such as Docker and Singularity, or even whole virtual machines, which provide a way to distribute computational environments to different machines and to execute within a given computational environment in a clean way. And finally, you need to actually manage the workflow itself. That is the actual execution of the analysis steps, beginning with the primary data and ending in the final results. And there's a plethora of tools available for doing this currently in the bioinformatics domain. Some of the popular ones include Toil, SnakeMake, and Nextflow. The uniqueness of Bionics is that it can solve all these three problems with one abstraction and one framework, which greatly simplifies the problem and increases the reproducibility guarantees from using the system. So to motivate this and to explain it a little bit, let's start with a simple example of a next generation sequencing analysis workflow, where we have done some sequencing and then we want to process it by doing an alignment, doing some sorting of the results from that alignment, and then doing some variant calling. Here is that process depicted as a graph, where arrows indicate that the output of one stage, like the alignment stage, is used in the subsequent step for sorting. So to be able to perform the sorting stage, you need to have done the alignment and have the results of the alignment stage. And to do the calling stage, you need to have the results from sorting. Of course, this depiction does not capture everything. So this is neither reproducible nor executable because we're missing some important components. First of which is data. So we have the reads that come off the machine, which are required by the alignment step, as well as reference databases that are required. For example, we need to choose a reference with which to align against. And that's not a piece of data we need. Other stages might require different bits of data that are obtained from different databases. For example, the variant calling stage may require access to databases to do annotation of those variants. So we can add the data to this graph, but it's still not yet reproducible nor executable and complete because we haven't specified software yet. So to do these stages of alignment, sort, and calling, we need to choose particular bits of software with which to do them. And some choices maybe BWA for the alignment, SAM-tools for doing the sorting, and Platypus for doing the variant calling. And so these are requirements for the steps. And yet this graph is still not complete because the software itself also has a bunch of requirements to be able to produce the BWA executable, for example. And these are things like Python, glibc, the various libraries that are involved. So in fact, there's a huge graph off to the left here of dependencies for the software itself, which need to be captured. If you were executing this in the traditional way and you wanted to provide this workflow to someone else for them to run and reproduce, then some of the technologies may turn towards using containers to capture the software component over to the left, perhaps distributing the databases required to run this workflow as a tarball, and then managing the steps in the middle or the actual analysis steps with the workflow manager. But let's take a step back and make an observation on this graph first. The first thing to observe is that there is no semantic difference between any of the arrows and any of the nodes. The alignment step requires some inputs to build and requires some build action to execute that build and produce an alignment. And similarly, the BWA, the build of software, is identical. It requires some inputs like the source code and its various dependencies. It executes some build action, which in the case of software is the compilation, and it produces some output, which in software cases is the executable. So there's no difference between software in this graph and alignment and analysis steps in this graph. Likewise, even the data itself is no different. So while data may not have any inputs or dependencies, it still has a build action because the data must be fetched from somewhere. So the build action, for example, would be to fetch a particular reference over network or from local storage in order to present it to the analysis stage. So all of these arrows and all of these nodes are the same. And therefore, if we have a good abstraction that we can use for modeling this graph, then we can build a workflow manager on top of it that captures everything. And that's exactly what Bionics does. It builds on top of Nix, which is a robust functional package management system. And because we treat everything the same, it allows us to manage both software, data, and workflows all within the one system, the one abstraction. Because it implicitly specifies its entire computational environment right down to the kernel level, it provides a high degree of reproducibility. Furthermore, as Nix is a functional language, it allows us to model workflow construction as simple function composition, which greatly simplifies the specification of various pipelines and workflows. And finally, Bionics does introduce a notion of typing of outputs in order to reduce errors. So Bionics will track the type of an output. For example, an alignment done with BWA would produce an output of type BAM, which also has an attribute that determines whether or not the BAM is sorted and in what sort order it is sorted in, if it is sorted. And by checking that the output types of various stages match the input types of subsequent stages, we can catch a lot of misspecification errors of workflows. So for example, in Bionics, you can't feed an unsorted BAM into a stage that requires BAM. That would be an error raised before the execution of the workflow. So you would catch this error upfront before any expensive computation is actually being conducted. So since Bionics builds on top of Nix, I have to explain a little bit about what Nix is. And in fact, it's a little bit confusing because Nix is overloaded and refers to two distinct things. The first thing Nix refers to is a simple configuration language, which is functional in nature. But you can think of it as being semantically equivalent to JSON, extended with a notion of having functions and a notion of having modules, so that you can write your configuration in multiple files and bring them all together with the application of functions. The second thing Nix refers to is a build engine that in a little bit of Nix jargon that realizes derivations. So in this context, a derivation is just a recipe for producing a build product that captures all its inputs and the actions that are required to produce the output. So by realized derivation, that just means to do a build and to produce a build product. And the build engine has all the features that you would expect a workflow system to have. It's robust, it can resume after errors, it handles errors properly, it logs everything, it supports parallelization, so on and so forth. Because Nix has been around for 17 years now, this build engine is quite robust and it has been used quite successfully in industry as well, so it's been well tested. Now, the important thing about Nix is that it controls side effects. So in this talk, a side effect is just going to be anything that we haven't explicitly captured in a derivation. So if an analysis stage or a build stage tries to refer to something that wasn't specified as an input, then that would be a side effect. And Nix prevents this from happening by running every build inside a sandbox where only the inputs that are specified are available. And this actually means that no network access is available during a build either, because network accesses are a form of side effect. Another unique aspect of Nix is that the output location is determined by hashing the derivation. So it's a function of the inputs that go in and the build recipe that produces the build. And therefore, the output location is unique to the specific build and a specific version of that build, which means that multiple versions of something can coexist simultaneously without causing an issue. Bionics itself is just a thin wrapper on top of Nix. What it provides is some functions for modeling workflow stages. And these are modeled as functions, which take some set of configuration options and some set of inputs and construct a derivation. It's also a library of wrappers for various bioinformatics tools, such as BWA and SAM tools, etc. And these are organized into a tree. So the top level of the Bionics tree refers to the tool, like bionics.bwa, bionics.samtools, and bionics.minimap2. And the second level of the tree refers to actions that you can undertake with that tool. So for example, there's a bwa.align and a minimap2.align, and therefore we can switch between alignments very simply by changing which tree we're referencing or indexing the align function out of. And finally, Bionics also provides some functions for Hpc execution. Since this is a common thing we need to do in the bioinformatics domain, there are some wrappers for Slurm and PBS, which allow builds to be submitted to a batch queuing system instead of executed directly in the build engine. So let's take a concrete example of a workflow that is specified in Bionics, which you see on the right-hand side here. So this workflow is a function. So this colon definition is of how you define a function in Nix. And it's a function from a set of configuration options, one of which is Bionics itself, a set of inputs, and some reference switch. If it's not supplied, it will just default to grch38 in this case. And given those parameters, it then defines a workflow which is variant calling over all the inputs where the inputs have been pre-processed. And we define pre-processing in this case to be just alignments followed by fixing mate pairs, sorting them, and marking duplicates. So inputs here is a list of inputs, and the map function just applies a function to all elements of a list and returns another list. So this map preprocess inputs just preprocesses all inputs and then passes them on to the platypus variant calling function. Now this pipe function is just a way to specify reverse function composition in Nix. So these are all actually functions, and pipe just composes them together into a workflow for us, which is quite nice. And on the left, we see a build graph that's generated, or partial build graph that's generated from applying this particular workflow to some concrete inputs. So the final stage of platypus variant calling which corresponds to this expression, is this node at the bottom of the graph labeled 1, corresponding to this code snippet 1 over here. In this graph, you can see all the stages that are managed by Bionics are indicated by these nodes without any decorations. Nodes with a rectangle decoration represent nodes that are software requirements, and nodes that are ovals represent data requirements for that stage. There's no semantic difference between any of these nodes. It's only a syntactic difference just to aid with interpretability of this. So that's how we define a simple workflow in this language. Now let's take a look at how you would define an individual component of this workflow, say this alignment stage, which is marked here on the left as number 2. So here's how a stage looks in the Bionics library. All of the stage, the wrapper functions are functions which take a set of configuration options, which is what you see at the top, a set of inputs, which is this middle bit here, and then produces a derivation somehow. So in this case, for the BWA alignment stage, our configuration options are the reference to align against, whether we want to convert the output to BAM or keep it as SAM, and Bionics itself. The inputs for the stage are either one or two files containing the reads, so for either single or paired end alignments. And then down the bottom, we create the actual build, or we declare the actual build recipe and what's needed to execute the stage. The build inputs parameter specifies software that we require to have in our environment for a successful build. In this case, we just reference BWA out of the Nix packages repository. And then optionally, we also require SAM tools that will produce a BAM output because we have to convert the SAM to a BAM with it. The build command just declares a small script which is executed to do the actual build. In this case, it just links in the reference to the current working directory, which is a temporary directory set up specifically for the build and in a sandbox, and then executes the BWA command to perform the actual alignment, given it details such as where the input files are found, the reference is aligning against, and optionally piping it through SAM tools to convert to a BAM, and then saving it to the output location, which is determined by Nix and exposed as an environment variable called out. So that's how one of the stages inside, or one of the wrappers, tool wrappers in Bionix library is declared, and it's relatively simple. Now let's take a look at how you would declare a bit of software inside the Nix system. For example, how do we declare this BWA if BWA did not exist in Nix packages repository, which is this node number 3 over here. So in this case, this expression was taken directly out of Nix packages repository, and the format of it is that it declares a function taking inputs and producing a derivation. So there's a standard build environment which contains things like a C compiler. There's something to fetch URLs from somewhere, and then there's a zlib requirement for BWA. And the derivation here is just specified by declaring where the source code comes from and what its build requirements are for. And by default, the standard builder will do the usual configure, make, install sort of process. But in this particular case, there is no make install for BWA. So an explicit install phase is declared as to how to install the output of make to somewhere. In this case, it just installs it to the output location binary directory as the BWA executable. All right, so I thought I would switch at this point and show a demo of that exact workflow executing. All right, so what we'll do is we'll spin up an Amazon AWS instance, a clean one, within which to execute our workflow. So that's what these commands are just doing. It's just spinning up a brand new instance, a T2 micro instance, and then SSHing into it. So we're now in a clean machine, which has absolutely nothing available to it apart from the Nix package management system. We just fetch the workflow from the Bionics repository. It's exactly the same as the workflow I just presented. So we do platypus variant calling over a bunch of inputs that have been pre-processed by alignment, fixed made, sort, and markups. Of course, this workflow is not complete because we haven't specified which inputs we would like to run it on. So we have to write an input specification which tells Nix which particular inputs to run it on. And again, we'll fetch that from the Bionics example repository. And here it is. So this just declares a couple of samples as input. Both of those are paired end reads, so they have 2 sets of FASTQ files associated with them. And in this simple example, the FASTQ files are just going to be fetched from GitHub because they're quite small directly. And same for the faster the reference as well. So we'll just fetch the reference from the GitHub repository, and then we'll import our workflow and pass it through our set of inputs and the references and the reference that we want it to run on. And that's sufficient to execute this workflow. So we can now ask Nix to to build it and tell it which Bionics to use, which version of Bionics to use to do this build. And as you can see, it goes off and it'll evaluate what's required, and you'll fetch bits of software that's required to do the build, and then execute the build and analyses doing the analysis steps as required. So it's already done the alignments, it's done the BWA mem alignments. Now it's done the platypus variant calling and produced some outputs. And it says down the bottom that 32 build artifacts were fetched from a cache. So these are likely software and bits of software that were required to do the build, and 19 were built. And if we look at the output of this, then we indeed can verify that a VCF file was produced as expected, with the variance, and for this particular example, with the variance that we expect as well. So there we have an example of taking a workflow specification and executing it on a completely fresh machine within the space of a couple of minutes. So we've been able to replicate that very, very easily. Okay, just for interest's sake, this is the actual dependency graph that was computed and then resolved to produce that particular example. So anyway, this is the actual dependency graph that was generated when evaluating that particular example. Right in the bottom left is our final stage, which is a platypus variant calling stage, and all this other stuff is requirements that need to be built in order to satisfy that. Right at the top here are actually things that you need to bootstrap a working Linux user environment. And that involves things like GCC, which is over here, Bash, which is over here, the Linux kernel, which is over here, Python 2 and Python 3 and things like that. So the system really does capture absolutely everything right down to the kernel level and what's needed to actually do the build. All right, so what about Hpc execution? With what I just demonstrated was execution on a fresh system, one machine. But of course, in bioinformatics, we frequently have to use computational resources which are exposed to a batch queuing system instead. And it turns out this is actually very easy to implement within the Bionics and Nix framework because it's very easy to take a derivation and override it and modify it producing another derivation. And therefore we can implement Hpc execution by writing a function that takes the derivation and modifies it by wrapping it in a small shell script to submit to a Slurm cluster instead of executing directly. And that's exactly what this little snippet of code to the left is. Now, I don't expect you to understand it, but this is actually the entire Slurm interface implementation. And as you can see, it's actually very, very short and very simple. And that's all that's really required to interface with the batch queuing system. Of course, as a user, you would never deal with this directly. Bionics exposes a more convenient functionality to request a submission of stuff to Hpc clusters instead. And the way it does this is by exposing a function at top level, like bionics.slurm, that takes a set of configuration options for the cluster or for the submission to the cluster, to the queuing system, and returns a complete Bionics tree where all of the tools have been overridden to submit to the cluster instead. So that means if you wanted to run your SAMTools sort on a Slurm cluster instead of directly on a node, you would just have to reference bionics.slurm.samtools.sort instead of bionics.samtools.sort. So it's quite convenient. And this construction allows us to easily shift workflows that are not explicitly written to run on a HPC to a HPC execution environment. For example, the workflow that we just executed, we could lift that into a Slurm queue by writing a small wrapper that instantiates a Bionics tree, which submits to Slurm with some default resource constraints. So let's say 24 nodes, 7 gigs of memory, and a wall time of 3 hours. And then we can import our old workflow and pass it through this tree, and it will execute on Slurm instead of directly on the build node. So you might wonder how this looks compared to Nextflow. And what I've done here is I've taken an example which is presented in the Nextflow documentation on the right, where I've done a simple workflow, and I've converted that to the equivalent expression in Bionics for doing the exact same workflow. So we've used exactly the same tools. We've done awk and rev, and we've reused them exactly the same in Bionics. So what does the workflow do? What it does is it takes a faster input file, and it splits every line into an independent file, and then it runs the reverse command on each file, which just reverses the characters in that file. So in effect, you are reversing every line of a faster file. In my opinion, I think it's actually easier to see in the Bionics version, because down the bottom we have the actual pipeline definition, which is again just simple function composition. So we can see that we are first splitting sequences, and then for each of those sequences we apply this reverse function, and we do that over our input. In Bionics, the reverse function you just define as a stage which takes the faster file and does the actual execution. Same for split sequences, we'll take a faster file, and then we run the awk command on that faster file and copy the output to the output location. So there are two things to note about the differences between Nextflow and Bionics here. First of all, in Bionics, we have to specify what software we're going to use. So for split sequences, we have to specify that we're going to use GNU awk. And for the reverse stage, we have to specify that we're using a utility out of the Linux utilities. This is because in Nix, if you don't specify your software, then it will not be available during build, because you must specify all input dependencies. In Nextflow, this is not the case. While you can specify containers that things should run in, it's not required, and Nextflow can simply assume that the software that you're going to execute is available in whatever computational environment you're executing the Nextflow pipeline in. So you can write a specification that's not complete in a sense that it doesn't capture the software. The second thing to note is that the Nextflow workflow or flow of data, to me anyway, is a little bit more opaque than in Bionics. So in Bionics, because we're using function composition to define our workflows, it's very explicit as to how the flow of data is. We split sequences, and for each of those we reverse. But Nextflow, each process is defined to manipulate some global arrays. So split sequences, for example, pulls an input faster file from some array called sequences, and then it outputs them into some array called records. And then the process reverse takes stuff from the array of records and then outputs it to standard output. So you kind of have to trace through these global variables of sequences and records to figure out what the actual flow between these processes are, which in my opinion is harder to understand than when you have function composition that defines the workflows. So Bionics is not just an academic dream. We've been using it in production at the WeHi on a reasonably large data set for quite a while now. We use it pretty extensively in the Stafford Fox rare cancer project. So in that project, we have over 100 samples. Most of them are whole genome sequencing. Some of them are whole exome sequencing. And at the moment, we're totaling about 20 terabytes of primary data. The workflow in this project is really complicated, so there's a lot of components to it. We do SNV calling, we do neoantigen prediction, structural variants, copy number calling, mutational signatures, and we also generate clinical reports which summarize all the results of the analyses. And we also manage the workflow specification, which is written in BioNix, in a Git repository so that we can exactly reproduce the versions of everything that we generate. And the reports are in fact embedded with the revision hash, which allows us to go back and recreate any particular part that we need to. We also have a continuous integration because this is a server. So because this is a research project, we're continuously changing our workflows. So we change our analysis technique, we change certain parameters with them, we add things to them quite frequently, and having a continuous integration allows us to keep the entire cohort consistent and up to date, all analyzed under the same workflow. And we have processed some samples within this project at Pawsey on a Slurm batch queue, and some samples at WeHi, both under a PBS system and a Slurm system. So because of the abstractions that we have used, it's very easy to change site and change queuing systems fairly transparently. So it doesn't require any modifications to the actual workflow itself, workflow specification itself, which has been very convenient to us. So in conclusion, Bionics is a unique approach to this system, to this workflow problem. And it provides a very high degree of reproducibility. So most of Bionics is actually able to do bit for bit perfect replication. Some tools are not bit for bit perfect, given the same inputs, but most of them are. And because it's all been unified under one framework, it obviates the need for the use of multiple technologies. So we don't need to use containers anymore, for example. And finally, the model for defining workflows as function composition really simplifies specification and management of these workflows. To just wrap up, I'd like to acknowledge my collaborators. So Leon and Tony, I did this work with them. So we came up with this and wrote a paper together. And I'd like to also thank the Papenfuss Lab, in particular Alan Rubin, who has been very helpful and I've had many discussions with, and also Jocelyn and Ramya for being very early adopters of the system and experiencing some of the early pain points. I'd also like to thank the Stafford Fox rare cancer team, within which we use this system quite heavily for our analyses, and also the Stafford Fox Medical Research Foundation for supporting me in my research. All right, thank you very much.
Thank you very much, Justin, for that great overview of Bionics and how it can be applied. We do now have time for questions. If you have a question or a comment, you can put that into the Q&A box that you see on your screen, and then Justin will be able to read them out and answer them. So I can see there's some coming in already there.
Yep, so the first comment is that Nextflow now has a second version, which is more functional in nature, and where the pipeline is specified more explicitly for improved clarity. So this is a great way to go in. I'm a big fan of modeling this as function composition because it really does simplify the construction of these. The second question is, how does compiling of the needed software scale when the required software gets quite large? That's a great question. So there is a global Nix cache, basically. So all of Nix packages is continuously built by a CI server, and the build artifacts are all available in the cache. So for most software, you do not need to actually compile it again, but it could compile it from scratch, but there's no need to because of the caching. you can just fetch it directly from the cache and avoid any lengthy software compilation. Another question, are there any tutorials for Bionics? That's a great question as well. There sort of is. There's a bunch of examples in the repository, so it's not really written in the style of something you should read end to end, but there are a bunch of examples of increasing complexity up until one example, which actually does run on a full whole genome sequencing dataset. So if you look at the repository in the examples directory, there's a collection of things available in there.
Justin, there's one question that's just come in through the chat box as well, rather than through the Q&A.
Ah, thank you. How many bioinformatics tools are available defined as Nix packages? How does it compare to Bioconda and biocontainers? That's a good question. I don't know off the top of my head how much is available in Bioconda and how much of that overlaps well with Nix packages. At the moment, I believe there's about 100 biology science packages available in Nix packages. And this is not including things like Bioconductor, because the whole of Bioconductor is available under Nix packages, for example. So that it would be interesting to try and figure that out. But I know that a lot of Bioconductor packages are present in Bioconda, for example. So it's not just a simple matter of counting. Got another question. How do you supply custom scripts to the workflow as a stage? So that would require writing. Where's my example gone? If you go back to the next flow. Here we go. These bits here, split sequences and reverse, are essentially defining the stage with the script in the build command. So these build commands are just little shell scripts that get executed. Now, if you already have a script that's not written in a Nix expression, that's not a problem. You can just write execute your current script in the build command, and that would execute the script. So you can refer to files that are available on your local file system very easily. What are the prospects of integration with cloud execution environments such as AWS Batch? How would that affect reproducibility given their reliance on Docker? I'm not familiar specifically with AWS Batch, but Nix, the build system, does have very good integration for remote building and farming out over build farms, because the Nix packages continuous integration server has to do exactly this, and that is actually hosted on a cloud instance. I don't think it's Amazon though. So it is very easy to do this on compute resources that you obtain from the cloud, but I'm not familiar enough exactly with AWS Batch as to how it would specifically integrate with that. Good question. Another question, is it possible to call Condo install packages as part of a workflow, or would we have to re-specify that package directly? Yes, it is possible. So Condo is available in Nix packages, so you can always invoke it to install some software out of Condo. So you can do that. There is a little bit of a, I guess you could say, as maybe some loss of reproducibility in theory, but you probably won't lose that much.
Okay, so thank you very much, Justin, for the webinar this afternoon, and thank you also for answering the very interesting questions that our participants have. Before leaving today, a short reminder that our next webinar is going to be on the 10th of December and will be presented by Dr. Anna Syme, and she'll be talking to us about plant genomics and chloroplast genome assembly using Galaxy Australia. Finally, just before we leave, we'd like to acknowledge our funding. The Australian Biocommons is enabled by ANCRIS funding via Bioplatforms Australia funding. Thank you so much for joining us today, and we hope to see you in another webinar again soon. Enjoy the rest of your day. Bye for now.